Hey there, and welcome back to the deep dive.
Hello.
Today, we're embarking on a mission, really, to truly understand something incredible, our sense of hearing.
Hmm.
You've given us this fantastic stack of detailed source material. It's basically a comprehensive physiological map of the whole auditory
It
system.
really lays it all out.
And our goal here is to take this map, unpack every single layer, and go deep enough that we genuinely grasp how sound travels from the air around us
Yeah.
all the way into the conscious experience in your mind.
Yeah.
Think of it as a deep dive to really master the mechanics of how we hear.
That's a great way to put it. It's one of those senses we, rely on constantly. But the machinery underneath, it's just astonishingly complex. We're talking about converting these subtle pressure waves in the air into meaningful neural signals the brain can actually interpret.
It's a whole chain of events.
It really is. Mechanics, fluid dynamics, electrical gradients, these incredibly intricate neural pathways. And honestly, getting into the details is the only way to truly appreciate the genius of this biological system.
Okay, let's unpack this then. Starting in the very beginning. How does sound even get into our heads in the first place?
Our journey starts, maybe obviously with the outer ear. That includes the pinna, the part you actually see on the side of your head, and the external acoustic meatus. That's the auditory canal.
Right, the ear canal.
Exactly. So sound waves, these little fluctuations in air pressure, they get collected, to some extent anyway, by the pinna, and then they travel down that canal.
Okay.
And at the end of that short tunnel, they encounter the first really crucial structure, the tympanic membrane.
The eardrum, the initial receiver.
Precisely. It's a delicate membrane, and when those pressure waves hit it, they cause it to vibrate. And this vibration is the critical first step, turning that airborne sound energy into actual mechanical motion.
And attached right to the inside of it is
Yeah.
one of those little bones.
Yes, attached directly to the inner surface is the handle of the first of three tiny bones. These make up the middle ear, the ossicle.
Ah, the famous trio. The malleus, the incus, and the stapes.
You got it. These are the smallest bones in the entire body, and they form this absolutely critical bridge.
A bridge from the eardrum
That's it.
inwards.
Exactly. The malleus, its handle, is attached to the eardrum, right? And it's rigidly down to the next bone, the incus. So as the malleus vibrates, the incus vibrates right along with it. No question.
Like they're locked together.
Pretty much. Then the other end of the incus articulates, it connects with the stem of the stapes. This whole chain acts together as a remarkably efficient system. Specifically, the malleus and incus function mainly as a single lever.
A lever needs a fulcrum, a pivot point. Where's that located?
Good question. Our source material describes the fulcrum as being roughly at the border where the tympanic membrane is suspended and the malleus is attached. So when an incoming sound wave pushes the eardrum inward, it pushes the malleus inward too.
Yeah.
And because of this lever action around that fulcrum, the incus gets moved, and that movement is transferred directly to the stapes.
And the stapes is key because its footplate or faceplate sits right against the oval window, which is like the doorway to the fluid-filled inner ear.
That's the absolutely crucial connection. So an inward movement of the eardrum and malleus pushes the stapes footplate inward on the oval window.
Right.
This inward push displaces the fluid inside the cochlea, pushing it forward. And conversely, when the eardrum moves outward, the stapes gets pulled outward, drawing the cochlear fluid backward.
So it's a direct mechanical translation. Eardrum vibration becomes fluid motion.
Perfectly put.
Yeah.
Direct mechanical translation of the eardrum's vibration into motion of the fluid inside the inner ear.
Fascinating. And there's a muscle mention that's always sort of tugging on this system, the tensor tympani.
Yes, that's a detail that often gets overlooked. But it's actually important for optimal performance. The tensor tympani muscle is constantly pulling the tip of the malleus handle inward. This keeps the tympanic membrane under a certain degree of tension.
But wouldn't a loose, floppy membrane vibrate more easily? Why would you want tension there? Seems counterintuitive.
It does seem that way, doesn't it?
Yeah.
But the source explains that this constant tension is actually vital. It ensures that vibrations hitting any part of the tympanic membrane, whether it's the center or way out near the edges, are effectively transmitted to the ossicular chain right where the malleus attaches.
Ah, okay.
If the membrane were completely lax, floppy vibrations hitting the periphery might just sort of ripple loosely without efficiently driving those bones.
So the tension kind of pre-tunes it for efficient transfer.
That's a good way to think about it. Pre-tunes the eardrum for efficient vibration transfer.
Okay, so we've captured air pressure waves, turned them into mechanical vibrations of these tiny bones. But the next step is going from the air-filled middle ear into the fluid-filled cochlea. That sounds like a massive challenge.
It is.
Sound travels easily through air, which is low inertia, but fluid is much denser, right? It resists movement way more. How do we avoid losing almost all the sound energy at that air-to-fluid boundary?
This is precisely where the ossicular system performs its absolutely critical function, something called impedance matching.
Impedance matching?
Yeah, think of it like this. Sound waves have a certain impedance, basically a resistance to motion, in air. The fluid in the cochlea has a much, much higher impedance.
Okay.
Trying to transfer sound energy directly from a low impedance medium like air to a high impedance medium like fluid is incredibly inefficient. Most of the energy would just be reflected back, kind of like light hitting a mirror.
Like shouting into a swimming pool from the air. Not much gets in.
Exactly that. Not much gets into the water. So the ossicles act as a biological impedance matcher.
Like a transformer making the connection efficient.
Exactly. They are beautifully designed to match the impedance of the air to the impedance of the cochlear fluid. Now, there's a point of clarity our source makes here that's important. While it acts like a mechanical transformer, the lever system of the malleus and ampexes doesn't actually increase the distance the stapes moves.
Oh, it doesn't?
No. In fact, the amplitude, the amount of movement of the stapes footplate at the oval window is only about 3/4 the amplitude of the malleus handle's movement at the eardrum. So the lever actually reduces the distance.
Okay, distance is reduced, but what about the force?
That's where the gain happens.
Okay.
The lever system increases the force of movement by about 1.3 times. But the much, much bigger factor in this impedance matching comes from the difference in surface area between the eardrum and the stapes footplate.
Ah, the size difference.
Huge difference. The tympanic membrane has a surface area of about 55 square millimeters. The stapes footplate pressing on that oval window is tiny, averaging only about 3.2 square millimeter.
Wow, 55 down to 3.2. That's a massive concentration.
It is. It's roughly a 17-fold difference in area. Now, if you combine that 17-fold area difference with a 1.3-fold force increase from the lever ratio,
Okay.
you get 17 multiplied by 1.3, which comes out to approximately 22 times more total force exerted on that small area of cochlear fluid via the staps, compared to the force exerted by the sound waves spread across the much larger eardrum area.
That's incredible. It's like concentrating all the energy from a large drum onto the very tip of a needle.
That's a perfect analogy. That increased force concentrated onto that tiny oval window is absolutely necessary to overcome the higher inertia of the cochlear fluid and make it vibrate effectively.
Right.
Our source highlights that this impedance matching is incredibly efficient for the frequencies most important for human hearing, sort of between 300 to 3,000 cycles per second, or hertz. It transfers somewhere between 50% and 75% of the incoming sound energy.
Which is pretty good.
It's really good. Without this system, most of the sound energy would simply bounce off the oval window.
What if the ossicles are damaged or don't move properly? Maybe, due to infection or some disease.
Yeah, our source clarifies that sound can still reach the cochlea in those cases, either by air vibrating the oval or round windows directly, or even vibrating the bony labyrinth itself. But the hearing sensitivity is dramatically reduced by about 15 to 20 decibels.
And what does that mean in real terms? 15 to 20 decibels.
Well, to put that in perspective, that's roughly the difference between hearing a medium-level voice clearly and barely being able to perceive it at all. It really shows just how vital that ossicular chain is for our normal, sensitive hearing.
This whole system sounds incredibly delicate, though. Does the ear have any kind of automatic protection against dangerously loud sounds?
Yeah, it absolutely does. There's a reflex mechanism involving those middle ear muscles called the attenuation reflex, sometimes called the middle ear reflex too.
Okay.
It gets triggered by loud sounds being transmitted through the ossicles. These signals go up to the central nervous system and then initiate muscle contractions back in the middle ear.
Which muscles are involved in that reflex?
It involves both the stapedius muscle, which is attached to the stapes and pulls it outward, away from the oval window, and the tensor tympani muscle, the one we mentioned earlier, pulling the malleus handle inward.
So they pull in opposite directions?
Yes. When loud sounds hit, these muscles contract simultaneously, pulling the ossicles in opposing directions.
And what does that contraction actually do to the ossicles?
It significantly increases the rigidity of the entire ossicular system. Think of it like bracing the chain, making it stiffer. This increased rigidity greatly reduces the efficiency with which vibrations are transmitted through the ossicles to the cochlea.
Does this happen instantly, like a flash?
No, there's a short latency, a delay, typically somewhere between 40 and 80 milliseconds. So a really sudden, extremely sharp, loud noise might still sneak through before the reflex fully engages.
Okay.
But for sustained loud sounds, it kicks in pretty quickly. And the key effect of this reflex is that it selectively reduces the transmission of low-frequency sounds, primarily those below about 1,000 cycles per second.
Ah, so it targets the low rumbles?
Exactly. The source indicates it can reduce the intensity of sound reaching the cochlea by 30 to 40 decibels in that frequency range.
30 to 40 decibels, that's like turning a loud voice down to a whisper.
It's a significant reduction, and its function is thought to be twofold. First, and maybe most importantly, protection. It shields the delicate hair cells and other structures inside the cochlea from the potentially damaging, excessively strong vibrations caused by very loud sounds.
Makes sense. And the second function?
Masking. By attenuating or turning down the low-frequency background noise in loud environments, it actually helps us focus on the sounds that are typically above 1,000 cycles per second.
Like speech.
Exactly. That's where a lot of the critical information in human speech is located.
Yeah.
So if you're in a noisy party, this reflex might help you concentrate on the conversation you're having by turning down the rumble of the crowd and maybe the bass from the music.
So it's like a biological noise cancellation system focused on the low end.
Pretty much, yeah.
And there was something mentioned about our own voice, too.
Yes, interestingly, this reflex is also activated when we speak. Collateral nerve signals are sent from the screech centers in the brain to the voice muscles, but they also activate the tensor tympani and stapedius muscles.
Huh. Why?
It seems to reduce our hearing sensitivity to the low-frequency components of our own voice, preventing it from sounding excessively loud to ourselves, especially through bone conduction or direct transmission within the head.
Speaking of bone conduction, is that another way sound can actually reach the inner ear?
It is. While air conduction through the eardrum and ossicles is definitely the primary route for normal hearing, the inner ear, the cochlea itself, is physically embedded within the solid temporal bone of the skull.
Right.
So vibrations of the entire skull can also cause the fluid inside the cochlea to vibrate.
Like when you put a vibrating tuning fork on your head.
Exactly that example. A tuning fork placed on your skull, or maybe an electronic vibrator on a bony prominence, like the mastoid process right behind your ear, can transmit sound directly to the cochlea through the bone, completely bypassing the middle ear.
But normal sounds in the air don't usually do that.
Not effectively, no. Normal loud sounds in the air usually don't have enough energy to cause significant hearing via bone conduction unless they are specifically amplified and directed to the bone using a special device, which is sometimes done in hearing aids for certain types of deafness, actually.
All right, so we've successfully captured sound, efficiently transferred its mechanical energy into the inner ear, the cochlea. This is where the real transformation happens, right? Turning mechanical vibrations into the electrical signals the brain understands.
This is the core of it, yeah.
What does the inside of this amazing coiled structure look like functionally?
Well, structurally, the cochlea is often described as this coiled system of tubes, like a snail shell. But if you were to kind of slice through it and unroll it functionally, like our source illustrates, you'd see 3 fluid-filled tubes running side by side for its entire length. There's the scala vestibuli, the scala media, and the scala tympani.
And they're separated by membranes.
Yes. The scala vestibuli is separated from the scala media by Reisner's membrane, and the scala tympani is separated from the scala media by the basilar membrane. And it's on the surface of this basilar membrane, sitting within the scala media, that you find the organ of Corti.
The organ of Corti. That's where the sensory action happens, the main event.
Precisely. The organ of Corti contains the hair cells, which are the actual sensory receptors of hearing. Now, structurally, you have these 3 chambers, but functionally, for just transmitting the vibrations, Reisner's membrane is so incredibly thin and flexible that it moves almost effortlessly with the fluid. As our source puts it, for the purpose of transmitting sound vibrations through the fluid, the scala vestibuli and scala media essentially act as a single chamber.
But Reisner's membrane must have another job then.
It does, an absolutely critical one, maintaining a unique fluid environment inside the scala media. And that special fluid is essential for the hair cells to function properly. We'll definitely come back to that.
Okay, so sound enters the scala vestibuli media via the stapes pushing on the oval window. What happens next with the fluid?
Right. When the stapes footplate pushes inward at the oval window, it displaces fluid in the scala vestibuli. Now, because the cochlea is this rigid, bone-enclosed space, That displaced fluid needs to go somewhere.
Yeah, it can't just compress.
Exactly. The compensation happens at the round window. That's another membrane-covered opening at the base of the cochlea right below the oval window. The round window membrane bulges outward when the stapes pushes inward.
Ah, like a pressure relief valve.
Sort of, yeah. It allows the fluid to move. So fluid flows from the oval window through the scalovestibular media. It goes around the very tip of the cochlea at a little opening called the
Helicotrema.
which connects the scalovestibula and scalotympani at the apex. And then the fluid flows back down the scalotympani towards that round window.
And as this fluid moves, it interacts with the basilar membrane that sits between the scalomedia and scalotympani.
That's the absolute key. The basilar membrane is not just a passive structure. It's a fibrous membrane, and it contains something like 20,000 to 30,000 stiff, elastic fibers. They stretch from the central bony core, the modiolus, out towards the outer wall.
Like tiny reeds.
Our source describes them exactly like reeds. They're fixed at their base in the modiolus, but their other ends are embedded in the membrane and are relatively free to vibrate. And critically, these fibers change dramatically along the length of the cochlea, from the base near the oval window all the way to the apex.
How do they change? What's different about them?
Well, their length increases significantly. They start at maybe 0.04 millimeters at the base and grow to about 0.5 millimeters at the apex. That's a 12-fold increase in length.
Wow.
Their diameter actually decreases. But perhaps the most important change is their stiffness. They decrease dramatically in stiffness by more than 100-fold from the base to the apex.
So short, stiff reeds near the beginning and long, much more flexible reeds near the end.
Exactly. And because of these changing physical properties, each point along the basilar membrane has a different natural resonant frequency.
Ah, okay.
The stiff, short fibers near the oval window at the base, they resonate best at very high frequencies. The long, limber, flexible fibers near the apex, they resonate best at low frequencies. And that low-frequency resonance at the apex is helped not only by the decreased stiffness, but also by the increased mass of the fluid column pushing down on it at that far end.
This sounds absolutely fundamental to how we sort out different pitches.
It is. It's the core mechanical sorter. But the fluid movement from the stapes doesn't just make one spot vibrate instantly. It actually creates a wave that travels along the membrane.
The famous traveling wave.
Precisely that. When the stapes pushes inward, displacing the fluid, it causes the basilar membrane near the base to bend downwards, toward the scalotympany and the round window. The elastic tension that builds up in those bent fibers then initiates a fluid wave. And this wave travels along the basilar membrane from the base towards the apex, towards the helicotrema.
Like a wave on a pond surface.
Yeah, our source uses analogies like that, or a pressure wave along arterial walls. It's a propagating disturbance.
And the behavior of this wave depends on the frequency of the sound, right? Linking back to those resonant frequencies.
This is where the magic happens, linking that traveling wave to the properties of the basilar membrane. The traveling wave actually starts relatively weak near the base where it's generated, but as it propagates along the basilar membrane, its amplitude grows. Critically, it reaches its maximum amplitude when it arrives at the specific point on the basilar membrane where the natural resonant frequency of the fibers there matches the frequency of the incoming sound wave.
So the wave gets bigger and bigger until it finds its sweet spot on the membrane, its resonant location.
Exactly. And what happens at that sweet spot, that point of maximum resonance? The membrane vibrates back and forth with its largest amplitude right there, and in doing so, it effectively dissipates the wave's energy. The wave essentially dies at that specific resonant point. It doesn't travel significantly further along the basilar membrane.
Right. Okay, so high-frequency waves find their resonant spot near the base where the fibers are short and stiff. They peak there and die out quickly.
Right.
Low-frequency waves have to travel much further, all the way down towards the apex, before they find their resonant spot on those long, flexible fibers, peak there, and then die out.
You've absolutely got it. A high-frequency sound creates a wave that peaks and dies out after traveling only a short distance. A medium-frequency sound travels maybe halfway before peaking, and a very low-frequency sound travels almost the entire length of the membrane, peaking near the apex.
Is the wave speed constant as it travels down the membrane?
No, and this is another clever detail. The source points out that the traveling wave actually moves quite fast along the initial portion of the basilar membrane near the base, but it progressively slows down as it moves further towards the apex.
Why?
It's linked to the changing elastic properties of the membrane, a high elasticity coefficient near the base, which decreases towards the apex.
And why is that speed change important? Does it matter that it slows down?
It's actually crucial for high-frequency discrimination. This rapid initial transmission near the base helps spread out and separate the vibrations caused by slightly different high frequencies within the first millimeter or so of the membrane.
I see.
Without this initial rapid phase, all the high-frequency waves would be so bunched up, so compressed at the very beginning, that we wouldn't be able to tell them apart effectively. It gives them room to separate.
So the combination of the basilar membrane's changing properties and this traveling wave means that for every specific sound frequency, there's a unique location along the basilar membrane that vibrates with maximum intensity.
This is the core principle. Our source has a figure, 53:5, that illustrates this beautifully. It shows how the pattern vibration is spread out, but it always has a distinct peak location that shifts predictably with frequency. This specific point of maximum vibration along the basilar membrane is the fundamental basis for how our auditory system initially sorts and discriminates between different sound frequencies. It's the foundation of the place principle.
And it's this vibrating basilar membrane with its frequency-specific peak that interacts with the organ of Corti, the sensory engine sitting on top of it.
Exactly. The organ of Corti sits right on the basilar membrane and, as we said, contains the hair cells. These are the biological transducers, the structures that convert the mechanical energy of the basilar membrane's vibration into electrical signals that the nervous system can actually process.
Okay, tell us about these hair cells, the actual receptors.
There are two main types. You have the inner hair cells, which are arranged in a single neat row along the basilar membrane. There are about 3,500 of them. They are the primary sensory transducers for conscious hearing.
Okay, the inner ones are key.
Then you have the external or outer hair cells. These are arranged in 3 or 4 rows, and they're much more numerous, about 12,000 cells.
And they're connected to the auditory nerve fibers.
Yes. The bases and sides of both the inner and outer hair cells are surrounded by this dense network of nerve endings from the cochlear nerve fibers. These fibers then bundle together to form the spiral ganglion of Corti, which is located in that central bony core of the cochlea, the modiolus.
Right.
Neurons in the spiral ganglion send their axons, maybe 30,000 fibers in total, out of the cochlea to form the auditory, or cochlear, nerve. And this nerve enters the brainstem at the upper medulla.
Okay, how does the vibration actually excite these hair cells? It must be related to those tiny hairs on top, the stereocilia.
It is, absolutely. The stereocilia are minute, stiff projections extending upward from the top surface of the hair cells. These stereocilia either touch or are actually embedded in the underside of the tectorial membrane. That's a kind of gel-like structure that lies above the hair cells within the scala media.
And how does the vibration cause them to bend or shear?
It's a really clever mechanical setup. The outer ends of the hair cells themselves are anchored in a stiff structure called the reticular lamina. And this lamina is supported by the triangular rods of Corti, which in turn are attached firmly to the basilar membrane fibers.
Okay, so they all move together.
Essentially, yes. When the basilar membrane vibrates upwards due to the traveling wave, this whole rigid unit, the basilar fibers, the rods, and the lamina rocks upward and slightly inward towards the modiolus.
Right.
When the basilar membrane moves downward, the unit rocks downward and slightly outward. Now, this inward and outward rocking motion causes the stereocilia, which are fixed at their base to the moving reticular lamina, but have their tips touching or embedded in the relatively stationary tectorial membrane above, to shear back and forth against that tectorial membrane.
Ah, so it's the relative motion that bends the hairs.
Precisely. It's this shearing force, the bending of the stereocilia relative to the hair cell body, that is the mechanical stimulus that excites the hair cells.
So the hair cells are excited by the bending of their stereocilia, caused by the relative movement between the basilar membrane unit and the tectorial membrane.
You got it. And the direction of bending is absolutely critical. Bending the stereocilia in one specific direction causes depolarization of the hair cell membrane, making it more likely to excite the nerve fiber connected to it.
Okay.
Bending in the opposite direction causes hyperpolarization, making it less likely to fire. This creates an alternating electrical signal in the hair cell that mirrors the vibration pattern of the sound wave.
We talked about inner and outer hair cells. The source says 90–95% of nerve fibers connect to the inner hair cells, so they must be the primary sensors, right?
Actually logical.
But it also notes that damaging the outer hair cells causes significant hearing loss. So what's the function of the outer hair cells if they don't seem to be sending as much direct information to the brain?
This is indeed a fascinating puzzle highlighted by our source. Despite the inner hair cells receiving the bulk of the neural connections and appearing to be the main transducers for conscious sound perception, the outer hair cells play a vital, though different role. Their proposed function isn't primarily detecting the sound signal itself,
Okay.
but rather controlling the sensitivity and the tuning of the inner hair cells, particularly at different frequencies. They essentially tune the receptive system.
They help refine the frequency sorting or maybe boost the sensitivity. How would they do that?
Well, there's strong evidence supporting this tuning role. There's a large number of nerve fibers that travel downward from the brainstem originating in the superior olivary nucleus back to the vicinity of the outer hair cells.
Signals going back to the ear.
Exactly. And stimulating these downward or efferent fibers can cause the outer hair cells to actually change length. They can shorten or lengthen. This change in shape could physically alter the mechanics of the basilar membrane or the relative movement between the basilar membrane and the tectorial membrane in that specific region.
Wow, so they can actively change the mechanics.
It suggests a very sophisticated feedback mechanism from the brain. The brain can send signals down to the outer hair cells to actively modulate the stiffness or the mechanics of the basilar membrane at specific locations. And by doing that, it could finely tune how sensitive the inner hair cells in that region are to particular frequencies, or perhaps sharpen the peak of the frequency response on the traveling wave.
So it's like the brain is adjusting the dials on its own input filters, right at the source.
That's a great analogy. The brain actively controlling the ear's sensitivity at the receptor level.
That's remarkable. Let's dig into the electrical side more. How does the mechanical bending of those stereocilia trigger an actual electrical signal?
Okay, this relies on the unique structure of the stereocilia themselves and also the equally unique fluid environment of the scala media. Each hair cell has maybe 100 stiff stereocilia. They're arranged in rows of progressively increasing length as you move across the cell surface away from the modiolus. A key structural component here is these thin filaments, described as tip links. They connect the very top of a shorter stereocilium to the side of the adjacent taller one.
Tip links connecting shorter to taller hairs. Got it. What happens when they bend?
Right. When the stereocilia bend towards the taller stereocilia, which happens when the basilar membrane moves upward and the reticular lamina rocks inward, these tip links get stretched taut, pulled tight. This mechanical tension physically pulls open about 200, 300 tiny cation-conducting channels. These channels are located near the tips of the shorter stereocilia. This pulling open is the critical mechanical to electrical transduction step.
Channels open, ions flow in. What ions are we talking about?
This is where that special fluid environment becomes critical again. Unlike most cells in our body, which are bathed in fluid high in sodium and low in potassium, the tips of the stereophilia are bathed in endolymph. And endolymph has a very high concentration of potassium ions, K+.
High potassium outside the cell.
Very high. So when these channels are pulled open, there's a rapid influx of positive potassium ions from the surrounding endolymph into the stereocilia and then into the main body of the hair cell.
Potassium influx causes depolarization. That's kind of the opposite of how many neurons work.
It is, and it's specifically because of that high potassium concentration in the endolymph, creating an inward driving force. This influx of positive charge depolarizes the hair cell membrane. This depolarization then triggers the opening of voltage-sensitive calcium channels, Ca2+.
Okay.
Calcium flows in, further boosting the depolarization. The cell then repolarizes, mainly by potassium ions exiting through different calcium-sensitive potassium channels.
So bending one way opens channels, potassium rushes in, depolarizes the cell. Bending the other way presumably closes them or allows other ions to flow, leading to hyperpolarization.
Precisely. This creates an alternating receptor potential in the hair cell, a fluctuating voltage that mirrors the frequency and amplitude of the basilar membrane's vibration. And it's this electrical potential change that triggers the release of neurotransmitters, likely the rapidly acting glutamate at the synapses with the auditory nerve fibers at the base of the hair cell, exciting those neurons and sending the signal onwards.
You keep mentioning this unique fluid environment and its importance. Let's talk more about the endocochlear potential.
Yes, this is a really crucial and unique feature of the cochlea. It plays a vital role in basically powering the hair cells. As we noted, the scala vestibuli and scala tympani are filled with perilymph that's similar to cerebrospinal fluid and is relatively neutral electrically, let's say around 0 millivolts.
Right.
But the scala media, where the hair cells and, importantly, their stereocilia are located, is filled with endolymph.
And the endolymph is different electrically.
Dramatically different. It's secreted by this specialized, highly vascular tissue called the stria vascularis, which is on the outer wall of the scala media. Endolymph, as we said, has a very high concentration of potassium ions and a very low concentration of sodium ions, the opposite of perilymph and most other extracellular fluids.
And this chemical difference creates an electrical potential difference.
Yes, and it's massive. There's a constant electrical potential of about plus 80 millivolts that exists between the endolymph inside the scala media and the perilymph in the scala vestibuli and tympani. The scala media is significantly positive relative to its surroundings.
Plus 80 millivolts.
That's the endocochlear potential, and it's generated by the continuous pumping and secretion of positive potassium ions into the scala media by that stereovascularis.
Okay, so the fluid around the stereocilia tips is sitting at plus 80 milliV. And the hair cells themselves have a negative resting potential inside, like most cells.
Right. The lower bodies of the hair cells are bathed in perilymph, which is around 0 milliV. And the inside of the hair cell itself is about radical 70 millivolts relative to the perilymph. But now, look at the electrical potential difference across the membrane at the very tips of the stereocilia, where they're sticking up into that plus 80 milliV endolymph.
Okay.
It's the intracellular potential relative to the endolymph potential. So, negative 70 millivolt inside the cell minus the plus 80 millivolts outside in the endolymph. That gives you a total potential difference across that tiny membrane patch of negative 150 millivolts.
It's minus 150 millivolts. That's enormous for a cell.
It truly is. And this incredibly large electrical gradient across the membrane, right at the stereocilia tips, is believed to be absolutely critical. It greatly sensitizes the hair cell. It provides this powerful electrochemical driving force for that influx of potassium ions when the mechanically gated channels are briefly pulled open by stereocilia bedding.
So it makes the response much stronger.
Significantly. It dramatically increases the hair cell's ability to respond rapidly and robustly to even the slightest vibration of the basilar membrane. It's what makes our hearing so incredibly sensitive. It's like having a high-voltage battery constantly charged and ready to power the signal transduction the instant it's needed.
So we've converted mechanical vibration into these fluctuating electrical potentials in the hair cells, powered in part by this unique endocochlear potential. These signals are then transmitted as nerve impulses up the auditory nerve. How does the brain then decode these patterns of impulses to perceive something as complex as frequency or pitch and loudness?
Right, that's the next massive layer of complexity, turning those raw encoded signals into conscious perception. We've already touched on frequency determination with the traveling wave and the basilar membrane's mechanics. The primary way the nervous system determines the frequency or pitch of a sound is based on where along the basilar membrane the maximal vibration occurred.
Because different frequencies cause peaks at different locations, low frequencies near the apex, high frequencies near the base. That place principle again?
Exactly. This spatial organization based on frequency, known as a tonotopic map, is remarkably maintained throughout the entire auditory pathway. From the cochlea, through all the brainstem relay stations we'll discuss, all the way up to the cerebral cortex.
A map of frequencies in the brain.
Essentially, yes. So specific groups of neurons in the brain are primarily activated by specific sound frequencies, corresponding directly to the location on the basilar membrane that was most stimulated. This is the fundamental place principle for frequency discrimination.
But the source mentioned a potential issue with very low frequencies, didn't it? Something below about 200 hertz. It seems the very tip of the basilar membrane, near the helicotrema, is stimulated by all frequencies below 200 hertz.
That's right. The peak broadens considerably at the apex.
So if they all stimulate the same general area, how does the brain tell, say, 50 hertz apart from 150 hertz using just the place? The location cue seems less precise down there.
That's an excellent point, and it points towards another proposed mechanism, particularly important for low-frequency discrimination, the volley or frequency principle. For sounds in the low to perhaps lower mid-frequency range, say from 20 hertz up to maybe 1500 or 2000 hertz, the nerve fibers in the cochlear nerve can actually fire bursts or volleys of impulses that are synchronized with the frequency of the incoming sound wave.
They fire in time with the wave.
They might have fire on every single wave cycle, especially at higher frequencies within that range, but the timing of their firing is locked to the cycle of the sound wave. So a 100 hertz sound might cause nerve fibers to fire in bursts 100 times per second.
Okay.
These synchronized volleys are transmitted up to the cochlear nuclei in the brainstem. And it's hypothesized that these nuclei, and perhaps higher centers, can actually distinguish the different frequencies based on the rate of these synchronized impulse volleys. This timing-based code could explain how we differentiate between very low frequencies where the spatial cues from the place principle become less distinct.
And there's evidence for this.
Our source notes that evidence comes partly from studies where the apical half of the cochlea is damaged, removing the main place cues for low frequencies. Yet some low-frequency discrimination ability is still retained, suggesting another mechanism like timing must be involved.
Okay, so pitch is primarily a where question for most frequencies, place principle, possibly supplemented by a when or how often question for low frequencies, volley principle. How about loudness? How does the brain know if a sound is just quiet or really loud?
Loudness perception is encoded in the auditory system in at least three interconnected ways, according to our source. The first one is the most intuitive. As the sound gets louder, the amplitude of vibration of the basilar membrane and the hair cells increases.
They move more.
Right. This causes the hair cells to excite their synaptic nerve endings at a more rapid rate. So, individual auditory neurons simply fire more frequently, more action potentials per second for louder sounds.
Faster firing rate signals more loudness. What's the second way?
Spatial summation. With increasing loudness and vibration amplitude, the stimulation on the basilar membrane doesn't just stay at a sharp peak, it spreads out slightly. It starts to activate a larger number of hair cells, not just those right at the peak frequency location, but also those on the fringes or the shoulders of the vibration pattern.
Okay, so more cells get involved.
Exactly. This means more auditory nerve fibers are activated simultaneously, leading to a greater total number of impulses reaching the brain over a given time.
Yes.
This spatial summation of impulses signals increased loudness to the brain.
More neurons firing means more loudness. Makes sense. And the third method mentioned.
This relates back to the outer hair cells again. Our source suggests that the outer hair cells might be primarily stimulated only when the basilar membrane vibration reaches high intensities. Their activation, when it occurs, could provide a specific signal to the nervous system indicating that the sound is definitely loud, perhaps contributing to the perception of very loud sounds.
So loudness is a combination of how fast individual neurons fire, how many neurons fire in total, and possibly a distinct signal from the outer hair cells at high levels. And how we perceive this change in loudness isn't linear, is it? It's compressed somehow.
Correct. Our perception of loudness follows what's known as a power law, which is actually similar to how we perceive changes in other sensory stimuli, like brightness or pressure. For sound, the interpreted sensation, the perceived loudness, changes roughly in proportion to the cube root of the actual physical sound intensity.
The cube root. That sounds like a very significant compression.
It is, and it's absolutely essential for our ability to hear effectively across the incredibly vast range of sound levels we encounter. Think about the dynamic range the ear handles. From the quietest, barely audible sound to the loudest sound before it becomes painful represents about a trillion-fold increase in sound energy, or maybe a million-fold increase in the physical amplitude of the basilar membrane's movement.
A trillion-fold? That's astronomical.
It is. Yet our auditory system compresses this enormous physical difference into a perceived change in loudness of only about 10,000-fold.
So a trillion-fold physical change becomes a 10,000-fold perceptual change. Why is that compression so important? What does it achieve?
This compression is absolutely vital. It allows us to discriminate subtle differences in sound level like 1 decibel, which our source says is about the smallest change we can barely distinguish in normal conversation ranges across an incredibly wide range of overall intensities. If our perception were linear, we'd either be overwhelmed by loud sounds or unable to hear quiet ones effectively. We could only operate well within a much narrower band of sound levels. This compression makes our hearing system robust and adaptable to drastically different acoustic environments, from a quiet library to a loud concert.
And this logarithmic relationship is why sound intensity is typically measured in decibels, right?
Precisely. Because of the extreme range and the somewhat logarithmic nature of our perception, using a logarithmic scale like decibels is far more practical and intuitive than trying to express intensities linearly with huge numbers. A bell represents a 10-fold increase in sound energy, and a decibel, dB, is simply 1/10 of a bell. A 1 dB change corresponds to about a 1.26 times increase in sound energy. This scale aligns much better with our compressed perception of loudness.
How sensitive is our hearing overall? What's the quietest sound we can possibly hear, and what's the full range of frequencies we can detect?
Our source includes a figure, 53/8, showing the threshold of hearing. That's the minimum sound pressure level required to just barely perceive a sound, plotted across different frequencies. And our hearing sensitivity isn't uniform across the board.
Where are we most sensitive?
We're most sensitive, meaning we can hear the absolute quietest sounds in the mid-frequency range, with a peak sensitivity usually around 3,000 cycles per second, or 3 kilohertz. At lower frequencies, say down at 100 hertz, a sound needs about 10,000 times more physical intensity to be heard at its threshold compared to a 3 kilohertz sound. We're much less sensitive to very low and very high pitches. And the overall frequency range. The full frequency range a young person with healthy hearing can typically detect is usually cited as being from about 20 cycles per second, a very low bass rumble, up to 20,000 cycles per second, a very high-pitched whine or hiss. However, our source makes an important point. This full 20 to 20,000 hertz range is really only accessible at relatively high sound intensities.
Ah, so at quieter levels, the range shrinks.
Yes. At lower loudnesses, say 60 decibels below the maximum reference level, the perceivable range might be much narrower, maybe only from 500 hertz to 5,000 hertz. And as many people experience, the ability to hear high frequencies often diminishes significantly with age. The range typically shortens, maybe to 58,000 hertz or even less in older individuals.
Okay, so we've generated these complex nerve signals encoding frequency and loudness right at the hair cells. These signals then travel up the auditory nerve. Where do they actually go in the brain, and how are they processed further along the way?
Right, the journey of the auditory signal into the central nervous system is quite a complex relay race, involving multiple processing stations. Figure 53-9 in our source maps this out. It starts with the nerve fibers from the spiral ganglion in the cochlea, forming the auditory nerve. This nerve enters the brainstem and makes its very first synapse in the dorsal and ventral cochlear nuclei, which are located in the upper part of the medulla.
The cochlear nuclei are the first stop. Got it. What's next?
From the cochlear nuclei, most of the second-order neurons, the next cells in the chain, cross over to the opposite side of the brainstem. They travel to synapse in the superior olivary nucleus, which is located in the pons. A smaller number of fibers actually stay on the same side and also project to the superior olive.
So that crossing over, that bilateral processing, starts very early on.
Very early. It means that information from each ear is already being processed by neurons on both sides of the brainstem right from this stage.
Where does the signal go from the superior olive?
From the superior olivary nucleus, the main pathway ascends up through the lateral lemniscus. That's a large tract of nerve fibers running through the pons and midbrain. Some fibers might synapse in the nucleus of the lateral lemniscus along the way, but many bypass it and continue upwards. The next major relay station is the inferior colliculus, located in the midbrain. Nearly all ascending auditory fibers synapse here.
The inferior colliculus? Then where? Getting closer to the top.
Closer, yes. From the inferior colliculus, the pathway continues upwards to the medial geniculate nucleus. This is part of the thalamus, which is like the brain's major sensory relay center. The medial geniculate nucleus is the final major processing and relay station before the signal reaches the auditory cortex. Essentially, all ascending auditory fibers synapse here.
Okay, the thalamus, and from there?
From the medial geniculate nucleus in the thalamus, the pathway proceeds via a bundle of fibers called the auditory radiation.
Finally reaching the ultimate destination, the auditory cortex.
Exactly. Terminating primarily in the superior gyrus of the temporal lobe, but our source notes it also extends onto the lateral temporal lobe, the insular cortex, and even part of the lateral parietal operculum. It's a fairly specific, but also somewhat distributed area.
Looking at that whole pathway from cochlear nucleus up to cortex, what are some key features that stand out about how the brain is handling this auditory information?
Well, that bilateral representation we mentioned is definitely crucial. Signals from both ears are transmitted along pathways on both sides of the brain, although the pathway carrying information predominantly from the opposite contralateral ear tends to be stronger or have more fibers.
And there are multiple places where signals cross over.
Yes, the crossing over isn't just at one point. It happens at multiple levels. In the trapezoid body, which connects the cochlear nuclei in the pons, and a chemisure connecting the nuclei of the lateral lemnisci in the midbrain, and also in a chemisure connecting the two inferior colliculi. This extensive crossing and bilateral processing is likely very important for functions like sound localization and hearing effectively in complex, noisy environments.
What else is notable?
Another important feature is the presence of collateral fibers that branch off the main ascending pathways, particularly lower down in the brainstem. Some of these collaterals project into the reticular activating system.
Ah, the brain's arousal system.
Exactly. It's a network that diffusely activates the entire brainstem and cortex. This is why sudden, loud sounds can instantly grab your attention, make you jump, and generally wake up your brain. They tap into this general arousal system. Other collaterals go to the vermis of the cerebellum, which is also involved in rapid motor responses, perhaps including reactions to sudden noises.
And that frequency map, the tonotopic map, is that preserved?
Yes. Throughout this entire pathway, from the cochlea all the way up to the cortex, that spatial organization, related to frequency, the tonotopic map, is largely maintained. Our source actually notes that multiple distinct tonotopic patterns seem to exist at different levels, like in the cochlear nuclei, the inferior colliculi, and several areas within the cortex itself. This suggests that this frequency map is absolutely fundamental to auditory processing at various stages.
What about the firing patterns of the neurons themselves along this path? Does the brain simply relay the signal like a wire, or is it actively processing it from the very start?
Oh, the processing begins surprisingly early. Even in the cochlear nuclei, the very first stop, individual neurons can fire at very high rates, maybe up to 1,000 times per second. And their firing rate is generally related to the loudness of the sound. Also, for frequencies up to about 2,000, 4,000 hertz, the firing of many of these neurons is often synchronized with the incoming sound waves, reflecting that volley principle potential we discussed for encoding low frequencies via timing.
Okay, but does that synchronization continue higher up?
Not really. As the signal travels higher up the brainstem tracts, that precise synchronization with the sound frequency is usually lost, except perhaps for very low frequencies, maybe below 200 hertz. And once you get above the inferior colliculi, even that low-frequency synchronization seems to mostly disappear.
So the higher up the pathway you go, the less the timing of individual neuron firing seems to directly relate to the sound frequency.
Right.
And the more the identity of the neuron that's firing, based on its specific place in the tonotopic map, becomes the main indicator of the frequency.
That seems to be the general trend, and it tells us something really important. The auditory system isn't just a passive relay wire. Information is being actively dissected, analyzed, and processed from the very first synapse in the cochlear nuclei onwards. This early processing is particularly critical for complex tasks like figuring out where a sound is coming from.
Which brings us neatly to the auditory cortex, the center for conscious perception. What actually happens when the signals finally arrive there?
Right, the auditory cortex, located mainly on that supratemporal plane of the superior temporal gyrus. This is where we consciously perceive sound, interpret its meaning, and make sense of complex acoustic patterns. It's not just one single uniform area, it's generally divided into primary and secondary or association areas. The primary auditory cortex receives the direct input projecting from the medial geniculate nucleus of the thalamus.
And the association areas.
The auditory association cortex gets excited secondarily, both by inputs from the primary cortex and also by projections from related thalamic association areas near the medial geniculate body.
And is that tonotopic map, the frequency map, still present in the cortex?
Absolutely. In fact, our source highlights the existence of at least 6 distinct tonotopic maps within the primary and association auditory cortical areas.
6 different maps of frequency. Why would we possibly need so many maps of essentially the same thing, frequency?
That's a great question. The thinking is that each of these different maps, or functionally distinct areas, is specialized to dissect out and analyze a specific feature or quality of the sound, all while using frequency as the organizing principle.
So they're looking at different aspects of the sound.
Exactly. One map or area might be primarily focused on discriminating pure tones and their precise pitch. Another might be more dedicated to analyzing the direction the sound is coming from. Others might be specialized to detect changes over time, things like the sudden onset of a sound, its duration, or rapid modulations in amplitude or frequency, which helps us distinguish between, say, a musical note, a noisy burst, or the complex patterns of speech. The cortex is essentially breaking down the sound into its constituent features across these different maps.
So the cortex isn't just registering, okay, this frequency is present. It's analyzing this frequency has this particular onset time, this duration, this loudness profile. Maybe it's changing pitch and it's coming from this specific direction.
Precisely. It's a much more sophisticated analysis of the sound's properties. And one really striking feature of cortical processing compared to lower levels is the sharpening of the frequency response.
Sharpening. How so?
While neurons lower down in the brainstem might respond to a relatively broad range of frequencies, kind of reflecting the spread of that traveling wave peak on the basilar membrane, neurons in the auditory cortex are typically much more selective. They respond strongly only to a much narrower band of frequencies. Their tuning curve is sharper.
How does that sharpening happen? What's the mechanism?
This sharpening effect is largely attributed to a process called lateral inhibition, which is a common mechanism used in many sensory systems to enhance contrast and precision.
Lateral inhibition. How does that work here?
Basically, collateral nerve fibers that branch off the primary excitatory pathway for a specific frequency actively inhibit the activity of neurons in adjacent pathways that are responding to slightly higher or lower frequencies. So stimulating one frequency strongly tends to suppress the response to nearby frequencies.
Ah, it enhances the peak by suppressing the neighbors.
Exactly. This contrast enhancement makes the response to the peak frequency stand out much more sharply against the background, allowing for finer discrimination of pitch. It's very analogous to how lateral inhibition in the visual system sharpens the perception of edges between light and dark areas.
And what about the auditory association areas? Are they involved in even higher level interpretation?
Yes, definitely. Neurons in the auditory association cortex don't typically respond just to simple frequencies or basic features. Their role is more about associating different sound elements with each other, like recognizing a sequence of tones as a familiar melody, or understanding the complex spectrotemporal patterns that make up speech sounds, or even associating sounds with information from other senses.
Connecting sound to other things.
Right. Our source notes, for instance, an overlap between the parietal part of the auditory association cortex and somatosensory area 2, suggesting potential links for integrating auditory and touch information.
So this is where we really make sense of sounds, give them meaning.
Exactly. The auditory cortex, particularly these association areas, is crucial for discriminating complex tonal and sequential sound patterns, like language and music. Evidence from animal studies, for example, shows that removing the auditory cortex bilaterally doesn't necessarily prevent the animal from detecting sounds or reacting crudely to them, but it completely eliminates their ability to discriminate complex sound patterns or subtle differences in pitch reliably. They lose the ability to interpret the sound's structure and meaning, and they often can't relearn these discriminations.
What about the effect of cortical lesions, specifically in humans? What does that tell us?
Damage to the auditory cortex in humans provides further, very specific insights. If both primary auditory cortices are removed or destroyed, it significantly impairs overall hearing sensitivity. However, if only one primary auditory cortex is removed,
Just one side.
yes, hearing in the opposite ear is usually only slightly reduced because of all those extensive crossovers in the pathways we talked about. But, interestingly, it does severely impair the ability to localize sound sources. Figuring out where a sound is coming from requires comparing and integrating the information process in both hemispheres, so damaging one side disrupts that process.
Okay. And lesions in the association areas.
Lesions that are restricted to the auditory association areas while leaving the primary cortex intact are particularly revealing about the role of interpretation versus basic detection. In these cases, hearing sensitivity and even basic tone discrimination might be perfectly normal. The person can hear the sound fine,
Ooh.
but they cannot understand the meaning of the sound. A classic example highlighted in the source relates to damage to Wernicke's area. This is located in the posterior part of the superior temporal gyrus, and it's a key part of the auditory association cortex involved in language comprehension.
Right, Wernicke's aphasia.
Exactly. People with lesions here can often hear spoken words perfectly well. Their basic hearing is fine. They might even be able to repeat the words verbatim. But they cannot understand the meaning of the words they are hearing and repeating. It underscores that conscious perception involves not just detecting the sound signal, but crucially, interpreting its significance based on learned patterns and associations stored and processed in these higher cortical areas.
That distinction is critical. Okay, one of the most vital pieces of information a sound can give us is its location in space. How does the auditory system figure out where a sound is coming from?
Sound localization, especially determining the direction of the horizontal plane, left versus right, relies primarily on comparing the input arriving at the two ears. Our brain uses two main cues for this.
Time lag and intensity difference, right?
Precisely. The first principle is the time lag, or the difference in arrival time, between when the sound reaches the ear that's closer to the source versus the ear that's farther away.
Makes sense.
If a sound is directly in front of you or directly behind, the time difference is essentially zero. But as the sound source moves off to one side, the sound wave arrives at the closer ear first, and there's a very slight delay before it reaches the farther ear. This cue, the interval time difference, ITD, is particularly effective and considered more exact for frequencies below about 3,000 cycles per second. It relies on very precise timing information in the brainstem.
Time difference for lower frequencies. What's the second cue?
The second principle is the intensity difference, or the difference in loudness of the sound levels reaching the two ears. A sound coming from your left will generally be slightly louder in your left ear than in your right ear. This cue, the interval level difference, ILD, works best at higher frequencies, typically above 3,000 cycles per second.
Why is it better at higher frequencies for intensity difference?
It's because at higher frequencies, the sound waves have shorter wavelengths. Your head actually acts as a more effective barrier or obstacle to these shorter waves. It casts an acoustic shadow on the side of the head, away from the sound source.
Ah, the head blocks the sound more.
Exactly. This shadow causes a noticeable reduction in sound intensity, or loudness, in the farther ear. At lower frequencies, the longer wavelengths tend to diffract or bend around the head much more easily, so the intensity difference between the two ears is much smaller and less reliable as a directional cue.
So time difference for low frequencies, intensity difference for high frequencies, mainly for telling left from right. What about knowing if a sound is above you or below or in front versus behind?
That's a bit trickier. Time lag and intensity difference are excellent for side-to-side azimuth localization, but they are much less effective for telling you about elevation, up, down, or distinguishing front from back, because the difference in timing or intensity between the two ears is minimal or ambiguous in those planes.
So how do we do that?
Distinguishing sounds in those dimensions relies primarily on the pinnae, the external ear structures, the fleshy parts we see on the outside. The complex shape of your pinna, with all its folds and curves, subtly modifies the incoming sound. It reflects and filters different frequencies depending on the angle the sound arrives from, especially in the vertical plane.
So the shape of our ears changes the sound slightly.
Yes, it imposes a direction-dependent spectral filter. Our brain learns, through experience, to associate these specific, subtle changes in the sound spectrum, the slight emphasis or attenuation of certain high frequencies caused by the pinna's shape with different vertical locations, or whether a sound is in front or behind. It's a learned skill based on these spectral cues provided by the outer ear.
Fascinating. Where does the brain actually start processing these directional cues? Does it wait until the cortex?
No, the initial analysis of sound direction begins remarkably early in the auditory pathway. Specifically, it starts in the superior olivary nuclei in the brainstem, the place we mentioned where signals from both ears first converge significantly. However, our source is careful to note that while the initial analysis happens down there, the entire neural pathway from these brainstem nuclei all the way up to the auditory cortex is necessary for the full interpretation and conscious perception of sound direction.
How do those superior olivary nuclei actually handle the time and intensity difference cues? Are there specialized circuits?
Yes, the superior olivary nucleus is highly specialized for exactly this. It's generally divided into two main parts relevant for localization. The lateral superior olivary nucleus, LSO, is primarily involved in processing those intensity differences, ILDs. It seems to contain neurons that receive excitatory input from the ipsilateral, same side ear, and inhibitory input from the contralateral, opposite side ear. The balance of this excitation and inhibition allows the LSO neurons to effectively compute the difference in loudness between the two ears and send a signal upwards that helps estimate direction based on that level difference cue, especially for high frequencies.
Okay, LSO for intensity. What about the time differences?
That's primarily the job of the medial superior olivary nucleus, MSO. The MSO has a fascinating and very specific neural circuit designed for detecting time lags, ITDs. It contains neurons that receive excitatory inputs from both ears, often arriving via precisely calibrated delay lines on their dendrites.
Delay lines.
Yes, the inputs from each ear travel along pathways of slightly different lengths before reaching the MSO neuron. As a result, a particular MSO neuron will fire most strongly only when the inputs from the left and right ears arrive simultaneously at its cell body. Because of the different path lengths, this simultaneous arrival only happens for a specific time difference between the sound reaching the two ears.
So different neurons are tuned to different time delays.
Exactly. Neurons at one end of the MSO might be tuned to respond best to a very short time lag, sound coming from slightly off-center. Neurons at the opposite end might respond best to the maximum possible time lag, sound coming from far to the side. And neurons in between respond optimally to intermediate time lags.
So the MSO effectively creates a spatial map based on the timing difference between the ears.
Precisely. Different populations of neurons within the MSO become maximally active depending on the precise interaural time delay. A sound straight ahead, 0 delay, activates one set of neurons. A sound from 30 degrees left activates another set. A sound from 60 degrees right activates yet another set. It creates this spatial pattern of neural activity within the nucleus that directly represents the horizontal location of the sound source based on those crucial time cues, particularly for low frequencies.
And this spatial map, this location information, is then sent up to the cortex.
Yes, this spatial orientation derived from the superior olivary nuclei, both the MSO's time map and the LSO's intensity map, is transmitted up through the pathway to the auditory cortex. The brain then determines the perceived sound direction by identifying which specific locus of neurons in the cortex corresponding to these maps is most stimulated.
And these directional signals might use separate pathways.
Our source suggests that these directional signals likely travel through a somewhat distinct pathway and terminate in a different area or areas of the cortex compared to the pathways primarily processing tonal patterns or pitch information. This again highlights that principle of feature extraction, how specific features or qualities of the sound, like its direction, are dissected out and processed separately, starting at different levels of the auditory pathway, like direction being computed initially in the superior olivary nuclei.
So we have all these signals going up from the ear to the brain for perception and interpretation. Is there also a system for the brain to send signals back down to the ear to influence what or how we hear?
Yes, absolutely. And it's a very important system known as centrifugal control or efferent pathways. There are descending or retrograde pathways at basically every level of the auditory nervous system, originating as high up as the auditory cortex itself and projecting all the way back down, ultimately reaching the cochlea.
All the way back down to the hair cells in the organ of Corti.
The main functionally understood downward pathway originates primarily from the superior olivary nucleus in the brainstem, the same nucleus involved in localization, and sends fibers directly back to make contact with the sound receptor hair cells in the organ of Corti, particularly the outer hair cells.
And what do these descending fibers do when they reach the hair cells?
These descending or efferent fibers are primarily inhibitory.
They reduce the activity or sensitivity of the hair cells.
Yes. Our source indicates that direct electrical stimulation of specific points within the superior olivary nucleus can cause significant inhibition of specific areas within the organ of Corti. This inhibition can actually reduce the hearing sensitivity of those areas to incoming sound by as much as 15 to 20 decibels.
15 to 20 decibels reduction right at the receptor level. That's quite substantial. What's the purpose of this inhibitory feedback loop? Why would the brain want to turn down its own input?
It's believed to be the neural basis for selective attention in hearing. This mechanism allows the brain to consciously, or perhaps subconsciously, focus our attention on sounds of particular qualities that are deemed important. Maybe sounds within a specific frequency range, or sounds coming from a particular location while simultaneously suppressing or rejecting other sounds that are not currently relevant or just distracting background noise.
So it's like the brain saying, pay attention to this, ignore that, like being able to pick out one specific voice in a crowded, noisy room, or maybe focusing on just the violin line in a complex orchestral piece.
Exactly that kind of scenario. As our source suggests, it allows us to attend to a single instrument within a symphony or a single speaker in a noisy environment by actively filtering out or inhibiting unwanted auditory information right at the very periphery of the system, modulating the response of the hair cells themselves. It's a remarkable example of the brain actively shaping and controlling its own sensory input based on context and intentional goals.
We've explored the incredibly intricate workings of normal hearing, but what happens when parts of this complex system are damaged or just don't function correctly?
Right, that leads us into the different types of hearing loss or deafness. Deafness is generally categorized based on where the underlying problem lies in that chain we've discussed. The first main category is typically called nerve deafness.
And nerve deafness involves the cochlea itself, or the neural pathways beyond it.
Yes, exactly. It's caused by some kind of impairment of the cochlea, including the hair cells, the auditory nerve leading from the cochlea up to the brainstem, or the central auditory nervous system circuits within the brain that process the sound information. If the damage is severe, particularly to the cochlea's hair cells or the auditory nerve fibers, this type of hearing loss is often permanent because these neural structures have very limited capacity to regenerate.
Okay, that's nerve deafness. What's the other main category?
The other category is conduction deafness. This is caused by problems with the physical structures in the outer or middle ear that are responsible for conducting the sound waves mechanically to the cochlea. So this includes issues with the external auditory canal, the tympanic membrane, eardrum, or that ossicular chain, malleus and datus stapes.
So the nerve part is okay, but the sound isn't getting through properly.
Precisely. In conduction deafness, the inner ear, the cochlea, and the auditory nerve are typically healthy and functional. This means that while normal air conduction of sound is impaired, sound can often still reach the functional cochlea via bone conduction, vibrating the skull directly.
How do doctors or audiologists diagnose the specific type and the degree of hearing loss a person might have?
They use a specialized instrument called an audiometer. It's designed specifically to assess hearing abilities systematically. An audiometer can generate pure tones, sounds of a single specific frequency across the typical range of human hearing, from low pitches to high pitches, and is very precisely calibrated.
Calibrated how?
It's calibrated so that a zero-intensity level sound setting at each frequency represents the average minimum loudness, the threshold that a normal hearing young adult can just barely perceive.
So if someone needs the sound turned up significantly above that zero level just to hear it, they have a hearing loss at that frequency.
Exactly. The audiometer has a calibrated volume control, usually measured in decibels of hearing loss, dBHL, relative to that normal zero threshold. If a person can only just hear a particular frequency when its intensity is raised, say 30 decibels above the normal zero level, they are documented as having a 30 decibel hearing loss at that specific frequency.
And they test this across different frequencies.
Yes, a standard hearing test typically involves measuring the hearing threshold at about 8 to 10 different frequencies spanning the main part of the audible spectrum, maybe from 250 Hz up to 8,000 Hz.
And this data is plotted visually on an audiogram.
That's right. The result is an audiogram, like the one shown in figures 5311 and 5312 in our source. It's a graph that plots the degree of hearing loss in decibels for each tested frequency. Crucially, the audiometer usually also has a mechanical vibrator that can be placed on the skull, typically on the mastoid process bone right behind the ear, to test hearing sensitivity, specifically via bone conduction.
Ah, so they test both ways.
Yes, comparing the results from air conduction using earphones and bone conduction using the vibrator on the audiogram is absolutely key to distinguishing between nerve deafness and conduction deafness.
What do those audiogram patterns typically look like for the different types?
Well, in nerve deafness, illustrated in figure 5311, the audiogram generally shows a similar degree of hearing loss when tested by both air conduction and bone conduction. This makes logical sense because the problem is either with the sensory receptor, the cochlea, or the nerve itself, so it affects both transmission routes more or less equally.
And are there common patterns within nerve deafness?
Yes, a very common pattern, particularly with age-related hearing loss, presbycusis, or noise-induced hearing loss, is deafness that is mainly for the high-frequency sounds. This is often caused by cumulative damage to the hair cells near the base of the cochlea, which, as we know, process high frequencies. Other patterns can occur too, of course. Sometimes deafness is mainly for low-frequency sounds, maybe from prolonged exposure to very loud, low-frequency noise, like from certain machinery or music, which can damage the organ of Corti differently. Or sometimes there's deafness across all frequencies, which can occasionally be caused by sensitivity to certain medications, like some specific antibiotics mentioned in the source, streptomycin, gentamicin, kanamycin, chloramphenicol.
Okay. And what does the audiogram look like for middle ear conduction deafness?
For conduction deafness, shown in figure 5312, the audiogram pattern is distinctly different. Bone conduction hearing is typically essentially normal, or at least much, much better than air conduction hearing. This tells you the cochlea and the nerve are likely working fine. However, air conduction, the sound traveling through the outer and middle ear via the eardrum and ossicles, is significantly depressed, showing a hearing loss across frequencies, often more pronounced at lower frequencies. There's a gap between the air conduction and bone conduction thresholds.
What typically causes this type of conduction deafness?
Common causes include things like chronic or repeated middle ear infections, otitis media, which can lead to fibrosis, scarring of the tympanic membrane, or damage or fixation of the ossicles. Another significant cause mentioned is a condition called otosclerosis.
Otosclerosis?
Yes. It's often a hereditary disease where there's abnormal bone growth in the middle ear. This leads to fibrosis and eventually calcification that prevents the ossicles, particularly the stapes, from vibrating freely as they should. In severe cases, the stape's footplate can become completely ankylosed, basically frozen or fused by this abnormal bone overgrowth to the edges of the oval window it sits in. This completely prevents the ossicular chain from conducting sound vibrations.
Can that condition, otosclerosis, be treated effectively?
Fortunately, yes, in many cases.
Yeah.
For severe stapes ankylosis, surgery is often very effective. The frozen staves could be surgically removed and then replaced with a tiny artificial prosthesis, perhaps made of Teflon or metal. This prosthesis bridges the gap, connecting the incus directly to the oval window again, effectively restoring the sound transmission path. Our source notes that this procedure, called a stapedectomy, can restore almost normal hearing in many cases of otosclerosis.
Wow. This deep dive has truly unpacked the stunning complexity of hearing. It's incredible to follow the journey from just a simple ripple in the air. We followed that energy through precise mechanical amplification and impedance matching by the ossicles into the generation of fluid waves in the cochlea, the beautiful frequency sorting by the basilar membrane's traveling wave, the transduction into electrical signals by specialized hair cells powered by that unique endocochlear potential.
Then the encoding of that information into neural signals that travel through this incredibly complex network in the brainstem and thalamus, with strategic crossovers and multiple processing stations.
Finally arriving in the cerebral cortex to be interpreted not just for basic pitch and loudness, but for complex patterns, for meaning, and for location in space.
It's just an astonishing chain of events when you lay it all out. The eardrum capturing the wave, the ossicles acting as that precise lever and amplifier, the basilar membrane and traveling wave sorting frequencies, the hair cells transducing with the help of that electrical boost, the nervous system dissecting features like pitch, loudness, location, all while the brain itself can actively reach back down and fine-tune the input via those efferent pathways.
We've really gone through the microscopic mechanics, the unique electrical environment, the physics of the wave propagation, the detailed neural pathways, and the higher-level interpretation in the cortex. It covers so many domains of science.
It really makes you stand back and just marvel at the sheer ingenuity of biological engineering. Every single part seems to have a specific, critical role to play in the overall function.
Absolutely. And thinking about that incredibly long and complex chain of events, all the delicate components involved, the membranes, the tiny bones, the fluids, the thousands of hair cells, the specialized neurons, and how precisely they all have to work together in concert for normal hearing.
It does raise a rather profound question, doesn't it? What does the sheer intricacy and, frankly, the numerous potential points of failure in a system as complex as hearing tell us about the necessary balance that must exist between fragility and resilience in highly complex biological machinery? How is it that this incredibly delicate system works so remarkably robustly for so long for most people, despite its apparent vulnerability at so many stages?
That is a powerful thought to leave with, thinking about that balance. Well, thank you for joining us on this incredibly detailed deep dive into the science of hearing.
It was my pleasure. Truly fascinating stuff.
We sincerely hope you, listening, walk away with a much deeper understanding and perhaps a newfound appreciation for the incredible, intricate process that lets you hear the world around you every single day.